HPT College, B.Sc Zoology, Mammalian Histology course, Chapter 3 Skin. The skin constitutes the main part of the integument and is remarkably complex, most versatile, and an adaptable organ that lends itself admirably to the maintenance of steady state homeostasis of the body. It wraps around the entire body and lines the oral and rectal cavity. It also serves uniquely as an important and effective biological barrier between the internal environment of cells and organs and the outside world. The skin is composed of an outer thin epithelial component, the epidermis, and an inner thick connective tissue component, the dermis or corneum, that forms its main bulk. The epidermis, its derivatives and nerve elements are formed from the ectoderm, whereas dermis, connective tissues and the vascular elements embedded in it are derived from the mesoderm. The total thickness of the skin varies from 1 to 4 mm. Under the dermis is a layer of connective and adipose tissues, the hypodermis, that connect the dermis to the underlying muscles and structure. Epidermis forms the outermost superficial part of the skin. It is composed of stratified squamous epithelium of keratinizing or cornified variety. The epidermis contains no blood vessels and tissue fluid. It contains end organ of sensory nerve. The lower surface of the entire epidermis is folded. A firm fixation of epidermis to the underlying dermis gives its appearance like that of an egg box. From the inside to the outside, the epidermis consists of 5 sheets or strata called as stratum basal, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. These sheets vary in microscopic appearance and are described in 2 major zones. The horny zone, it is the superficial portion of epidermis and is composed of 2 layers of cells which have lost mitotic potential and represent progressive stages of cornification. There are 2 layers, stratum lucidum and stratum corneum. Second one is a stratum corneum, that is called a horny layer. It is the most superficial layer of epidermis. It is composed of clear, dead, and very large flattened scale-like cells arranged in 25 to 30 layers. They have no nuclei. The protoplasm in these cells is completely changed into a horny substance known as a keratin. It is this mantle of dead tissue called the horny layer that serves as the principal waterproof shield of the body. Keratin is made up of microfilaments which fill the cytoplasm. All other organelles have disappeared. Keratin is tough and resilient and thus contribute to protective qualities of epidermis. The stratum corneum has a lower boundary in the form of a membrane called the stratum conjunctum, which perform an important protective function. The most superficial layer of stratum corneum show little cohesion and are constantly being exploited or shed by wear and tear of and get exposure to the environment. Because of this tendency, the superficial layer of stratum corneum is also termed as stratum disjunctum. Stratum lucidum is clear cell layer or hyaline layer. It is absent in thin skin, but in thick skin it is present as a brilliant, structureless, and slightly acidophilic thin zone of transition between granular and cornified layers. It is composed of 1 or 2 layers of extremely flattened unnucleated cells. Their cell boundaries are quite indistinct and the cell components are not evident. Carinohyaline granules have disappeared and are replaced by semi-liquid droplets of elidine. which represent the second stage in the formation of keratin. Stratum granulosum, granular cell layer. In thick skin, this grainy layer is composed of one of the four layers of flattened, horizontally arranged spindle-shaped cells. But in thin skin, it is composed of only scattered cells forming an incomplete layer above the stratum spinosum. Their cytoplasm is filled with prominent, darkly stained, irregularly shaped keratohyaline granules. Keratohyaline serve as a precursor in the definitive process of keratinization. Granulosome cells usually show nuclei in various stages of degeneration. Stratum spinosum or stratum acantholeum, also called as a prickle cell layer, is situated above the basal layer. It is composed of 2 to 10 layers of polyhedral or polygonal cells with a distinct round nuclei and abundant cytoplasm. The cells are larger than those of the basal cell layer. Their cell membrane possess prominent short spine-like protoplasmic process by which they are joined together. These cells divide mitotically, but the mitotic activity is not as high as in the cells of basal layer. As the cells move outward towards the outer layer of epidermis, they lose water and gradually begin to flatten. Most of their organelles also start disappearing. Stratum basal or stratum germinativum, basal cell layer. It is the deepest layer of epidermis that rests on the basement membrane. It is formed of a single row of cuboidal or short columnar cells with oblong nuclei. The axes of cells are arranged approximately perpendicular to the basement membrane. This layer serves as a source of stem cells for other layers. Its cells reproduce rapidly by mitosis and continuously move upward toward the surface to replace those which are constantly being lost. As the cells move towards the surface, they engage in the synthesis of an intracellular protein called keratin. Evenly scattered among the cells of stratum basals and lowest layer of stratum spinosums are to be found two types of special cells, melanocytes and cells of Langerhans. Cell types of epidermis. There are four types of cells have been described in epidermis as keratinocytes, melanocytes, Langerhans cells, and Merkel cells. Keratinocytes or keratinization. Keratinocytes are the main and most predominant cells of the epidermis. Most predominant cells of the epidermis involved in a complex process of keratinization. The cytomorphology of these cells constantly changes as they move from the basal to the superficial layer of the epidermis. These facts constitute the basis of naming the various layers of epidermis. Keratinization. The main role of keratinocytes is to produce keratin, a fibrous protein that covers the skin with protective sheaths. The life cycle of the cell shows that the epidermis is continuously renewing itself to make good for the loss of cells at the surface. Death is thus the ultimate goal of every epidermal cell. This is achieved in an orderly manner by the process of keratinization, which may be regarded as a form of cell differentiation or cytomorphosis involving proliferations, a synthetic activity, differentiations, and a degenerative stage. Second is a melanocyte. The melanocytes constitute a small population of cells which arises from the neural crest of ectoderm. These are specialized cells responsible for the production of pigment melanin, which has a protective function. Melanocytes are distributed among the keratinocytes throughout the basal and some spinous layers of epidermis. These are spider-shaped cells with few to numerous long tentacle-like cytoplasmic processes that extend between the adjacent epithelial cells of stratum spinosum and terminate in expansion. Electromicroscopically, the melanocytes are characterized by presence of melanin granules of melanosomes. Melanin produced by melanocytes and stored in melanosome is either injected directly into keratinocytes by the melanocytes or the melanin-containing cytoplasmic processes of melanocytes are actively ingested by the keratinocytes of a basal and spinous layer. The prime function of pigment is to provide a shield to the underlying tissues by absorbing the ultraviolet radiations. The melanocytes also impart color to the skin and hair. The color is also determined by the blood in the capillaries of dermis. Merkel cells. These cells are thought to have been derived from the neural crest. Merkel cells are thought to have been derived from neural crest. They move into the epidermis from the dermis during the embryonic life and tend to lie in close contact with the sensory nerves ending in a near the stratum basals of epidermis and are also found scattered among the keratinocytes of epidermis. These are distinctive, clear cells which bear resemblance with those of the stratum spinosum and unlike the Langerhans cells are attached to the neighboring epithelial cells by desmosomes. Electromicroscopically, they contain numerous granules with dense cores, which are unique among epidermal cells. These membrane-bound granules are clustered on the side of Merkel cells that faces the underlying dermis. This is the dermal face which is in close contact with the nerve ending. The function significance of these cells is still obscure, but due to their deep contact with the nerve ending in epidermis and dermis, they may play a role in sensory process, such as touch receptions in many mammals. The granules resemble those present in catecholamine and polypeptide producing cells of paraganglia and adrenal medulla. Langerhans cells. These cells are present among the stratum spinosum. Although their presence elsewhere in the epidermis is not clear, they are satellite or dendritic cells which extend cytoplasmic process into the stratum spinosum. At electromicrograph level, they shows Langerhans granules which constitute the distinctive feature of these cells. The granules are characteristically membrane-bound rod-like structures often found in close contact or continuous with the cell membrane. Langerhans cells are reticuloepithelial cells and serve as epidermal phagocytes. Dermis, also called as a corium, underlying the epidermis, is a tough, flexible and elastic layer of dense, irregularly arranged fibroblastic connective tissue that constitute the main bulk of the skin. The dermis is of mesodermal origin and developed from mesenchymal cells. It contains various skin appendages, neural and vascular elements. The cellular elements of the dermis are relatively spare, most abundant being the fibroblast. Other includes melanocytes, mast cells, fat cells, and migratory leukocytes. The dermis is generally divided into an outer superficial papillary layer and the inner or deeper reticular layer. Stratum papillary or papillary layer constitute about one-fifth of total dermis and is not sharply demarcated from the reticular layer. This layer is characterized by a loose and irregular arrangement of thin collagen fibers embedded in a considerable amount of ground substance. The interface between dermis and epidermis is known as dermoepidermal junction. It presents a scalloped appearance like that of an egg box. In this arrangement, the papillary layer forms conical eminence or dermal papillae. This layer has a rich capillary network in the form of a loop arranged perpendicularly to the skin surface. This loop serves to nourish the epidermis which is otherwise avascular and also act as heat regulator. Nerves in organ, particularly the tactile bodies, are located in some of the dermal papillae. Second layer is a stratum reticulare or reticular layer, in much more dense than the papillary layer and consists of large, coarse bundles of collagen fibers aligned parallel to the surface and embedded in a lesser amount of ground substance. Because of toughness, this layer forms the basis of making of leather goods in some animals. There are fewer cells than in papillary layer and some elastic fibers may also be present. Blood vessels and nerves as well as sweat glands and hair follicles are found in this layer. Smooth muscles of erectors pilorum are also present. Apart from these two layers, dermis also consists some epidermal accessory organs like hair follicles and glands, also contain cells of connective tissue proper, and they have some blood vessels, lymphatic vessels, nerves for communication with other organ system. Hypodermis or subcutaneous layer of skin. The deeper part of the dermis borders on the subcutaneous tissue which is referred as the hypodermis. It is not a part of the skin proper but only serves to support and attach the skin to the deep fascia, muscles and bones. The loose constructions of this layer permits movement of skin in relation to this underlying structure. It is composed of loose connective tissue in honeycomb arrangement with numerous fat cells. In a deeper part of this layer, an abundance of adipose tissue may be deposited in the form of a fat pads termed as the panniculus adipose. There is no clear line of demarcation between hypodermis and the dermis other than this fatty deposition. Portion of sweat glands, hair follicle, blood vessels, lymphatic vessels, nerves and some sensory end organs lie in this layer. These layers serve as a buffer, as a storage structure for food and water and as a thermal insulator. The epidermis is avascular but the dermis contains extraordinary dense network of blood vessels like arterial supply, venous drainage and lymphatic drainage is seen in a hypodermis layer.